Hi everyone, my name is Guadalupe Martinez, and today I'm going to talk about ethical issues on human therapeutic and reproductive cloning. Over the years, science has made great advances on research, such as cloning, which was initially developed with animals. The Boulder in 2008 mentioned that cloning is a replication of a hereditary copy of a DNA sequence or the entire genome of an organism. It is characterized by the advances used to create an exact genetic duplicate of another cell, tissue, or organism. The creation of a replica can be created in two ways, naturally or artificially. In laboratory, by twinning or embryo splitting, an early embryo is divided in vitro so that both parts, when transferred to the uterus, can return into hereditary independent organisms identical to each other. The first cloning occurred with frogs by scientists Robert Brinks and Thomas Kings. But in 1997 was the year of the clone science, the illustration and announcement of the birth of Dolly. The sheep clone from an adult sheep in Scotland appeared on magazine covers and Science on December 19, 1997, proclaimed that Dolly was the great advance of the year. Because of this, in all media, they were more fervent in their advertisement about Dolly. McLean, 1999. Through cloning, identical copies can be created and it can also help solve some disease. This technology is revolutionizing by creating the ability to progress in the therapeutic and reproductive areas. But what are the ethical issues in human therapeutic cloning? Due to recently found findings that unfortunately are not closely related to cloning, the possibility of curing disease by performing transplants not of complete organs but of cells through so-called cell therapy. This seems a good alternative for certain diseases that are a consequence of the malfunction of a well-defined population of cells, replacing diseased cells with healthy ones without the need to transplant the entire organ. Kefori in 2007 mentioned that somatic cell nuclear transfer in the medical reproduction setting is of sufficient magnitude for clinical examination and application. The creation of human heritage, animal cases, and cures through cell replacement in regenerative medicine. The fusion of therapeutic reduplication and gene therapy shows potential benefits in redeeming a patient for a genetic mutation that results in loss of function. Cloning as a therapeutic from ensures a promised future for medicine and for all human beings who require some treatment, considering this an option to solve the discomfort. Therapeutic cloning applying cell replacement therapy can generate numerous variety of tissues, for example, osteoblasts to reduce osteoporosis and revitalize the spinal cord and after trauma. Kiefer in 2007. On the other hand, ethical dilemmas in human reproductive cloning. The creation of Dolly brought with it numerous pros and cons, wondering if this could be developed in humans, because for society these technological advances would have great significance. Narvari in 2013 mentions that it may play an important role in the fact that after the creation of Dolly, most governments committed to adopting reassuring roles prohibiting this type of human procreation, although the regulations are not complete. Reproductive cloning would also be a kind of method to create an embryo, a child, or human being. Arguments debate and question the idea of human cloning for reproductive purposes, such as religious and moral, since they are based on the doctrine of deep personal thoughts and an embryo is a human being, which should be created between two humans and reproduced naturally, resulting in the rejection of manipulation and experiment on human embryos. Science involves an accelerated rate and is capable of remedying challenges. Advances in genetics and molecular biology continue to surprise society since the cloning of Dolly the sheep was the first animal cloned more than 20 years ago, marked a new stage in the investigation. Due to reproductive process in animals, reproductive technologies have been promoted and developed for animals and humans in three ways. The first one is artificial insemination, second, superovulation and embryo transfer, three, in vitro fertilization procedures. Current progress and advances in stem cell studies have given efficient opportunities for society, although they have shown scientific and ethical difficulties. They have promising future in regenerative medicine in the treatment of genetic conditions. Advantages and global reach as it could provide numerous medical advances. Cloning with a therapeutic approach focuses on generating stem cells to generate new tissues, which could help organ transplant and result in the efficiency of new treatments for conditions. On the other hand, human reproductive cloning, if in the future it well considered ethical, acceptable, and safe, could have a great impact worldwide that could produce genetically compatible organisms for transplant, which could help to the shortage, thus alleviate the long wait list for transplants. Another great impact that cloning technology can have is to preserve endangered species by creating clones from preserved tissue samples. About genomes is that people cannot similarly create them, but in a few years, therapeutic cloning will provide the ability to replace organs, heal nerves, cells, tissues, and other health benefits. However, the power to perform human cloning for reproductive is admitted with shock, concern, and even prohibition possibility by the International Committee for Monitoring Assisted Reproductive Technology and the World Health Organization. 2003, a group of medical experts and scientists petitioned the United Nations for a ruling by the International Court of Justice to determine human cloning as a crime against humanity, since this means conducting experiments with human beings.